Hello friends, assalamu alaikum. Today we will discuss a new drug carrier system which is called the Niosomes. A Niosome is non-ionic surfactant based vesicle. Niosomes are made up of a non-ionic surfactant. So what is surfactant? Surfactant are the substances which reduce the surface tension between two phases, and non-ionic means that they don't have charge. So Niosomes are made up of non-ionic surfactant, and they look like a vesicle or spherical in shape. So you can see here in this diagram, this is a spherical shape vesicle, and this is a Niosome. So how we obtain the Niosomes? So Niosomes are obtained on the hydration of synthetic non-ionic surfactant with or without incorporation of cholesterol. So we can get the Niosomes when we add water to the synthetic non-ionic surfactants, and sometimes we also add the cholesterol. So Niosomes are the drug carrier system made up of non-ionic surfactant, and they look like a vesicle or sphere in shape. Niosomes are used as carrier for hydrophilic, lipophilic and amphiphilic drugs. So with the help of Niosomes we can deliver drug like hydrophilic, lipophilic and amphiphilic. So hydrophilic drugs are those drugs which have affinity towards water. Lipophilic drugs are those drugs which have affinity towards lipids, and amphiphilic drugs are those drugs which contain both portion, hydrophilic portion and lipophilic portion. So with the help of Niosome we can deliver these three types of drug. Size of the Niosome is from 10 nanometer to 1000 nanometer. In the Niosome, the central area is the aqueous compartment. Here we can incorporate the water soluble drug, and this area is called the non-aqueous non-ionic surfactant bilayer. So here we can incorporate the lipid soluble drug. So what are the advantages of the Niosomes? With the help of Niosomes, different solubility drugs can be delivered to the body. Second one, controlled release of drugs we can obtain with the help of Niosomes. Third one is osmotically active and stable. Niosomes are actually osmotically active. It means that water can penetrate into the Niosomes, and it is also stable. The fourth one is increase the solubility of entrapped drug. The drug which we incorporate in the Niosomes, so we can increase the stability of that drug. The fifth one is no special requirements for the storage and handling. The sixth one is Niosome can be used via oral, topical and parental route. So we can administer the Niosome orally, topically and parentally. Seventh one is low production cost, so it is cheap as compared to other carriers like liposomes. And eighth one is less toxic. It is less toxic because it is non-ionic in nature. Methods of Niosome preparation. So how we can prepare Niosome? So the first method is ether injection method. As the name indicate that something is injected here. So the first step in ether injection method is that we will prepare mixture. We will take a beaker and we will add ether. Then after this we will add the surfactant plus lipid. So we will get a mixture. Then we will take a protein gauge injection and we will fill this injection from the mixture. Then in the third step we will prepare aqueous solution of drug. We will take a beaker, we will add water, then we will add a drug. So this will be aqueous solution of the drug, and this aqueous solution of the drug will be heated on the water bath at 60 degrees centigrade. Then we will inject the first one mixture in the aqueous solution of the drug. So after this, in the fourth step we will evaporate the ether, and we will get the Niosomes. So in ether injection method, first we will prepare mixture of the surfactant, lipid and ether. Then this mixture will be filled in the protein gauge injection. Then we will prepare the aqueous solution of the drug and we will inject that mixture in the aqueous solution of the drug. In the fourth step we will evaporate the ether and we will get the Niosomes. So the second method is thin film hydration method. In this method, first we will take a flask. In the flask we will take organic solvent, then we will add the surfactant and cholesterol. So we will get a mixture. Then in the second step we will evaporate the organic solvent from the mixture using a rotary evaporator. This is a rotary evaporator, and with the help of water bath at 60 degrees centigrade we will evaporate the organic solvent. So after evaporation of the organic solvent, a thin layer of surfactant plus lipid will form on the wall of flask. So when we evaporate the organic solvent, we will get a thin film of the surfactant and lipids on the wall of the flask. And in the next step we will prepare aqueous drug solution. We will take water and we will add the drug, so we will get aqueous drug solution. This aqueous drug solution will be added to the flask, so we will get a multi-layer Niosomes. So the third method is multiple membrane extrusion method. So what is extrusion process? Extrusion process is a process in which we pass something from a narrow hole. It's called extrusion, and the product we get is called extruded product. So in this method, first we will prepare mixture of the cholesterol and surfactant in the chloroform. Then we will evaporate the chloroform and we will get a thin film like this. We will get a thin film of the lipid and cholesterol. Then we will add the aqueous drug solution. After adding the aqueous drug solution, we will get a suspension, and that suspension is passed from the polycarbonated membrane. So when we pass that suspension from the polycarbonate membrane, so we will get the Niosomes, and that Niosome will be called extruded Niosomes. You can see here in the diagram, so this is the suspension we prepared and this is the membrane, and when we pass this suspension from the membrane, so we will get the Niosomes, and these Niosomes are called extruded Niosomes. So the fourth one method is the reverse phase evaporation method. In this method, first we will prepare drug aqueous phase. We will add the drug to the water, so this will be drug aqueous phase. We will also prepare the organic phase. We will take a beaker. In the beaker we will take ether or a chloroform. After this we will add 50% surfactant and 50% cholesterol, so this will be the organic phase. After this we will add both the solution and we will sonicate at 4 to 5 degrees centigrade. So sonication is a process in which we use the sound waves to agitate or to mix the particle or to mix the solution. So after sonication, a clear gel will be formed. To that gel we will add a small phosphate buffer solution, then we will again sonicate. In the fifth step, we will evaporate the organic phase at 40 degrees centigrade. In the sixth step, we will get the viscous Niosome suspension. So if we want, we can dilute the viscous Niosome suspension with the help of phosphate buffer, heating on the water bath at 60 degrees centigrade for 10 minutes. So application of the Niosome, the first one is drug targeting. With the help of Niosome, we can target a specific site. Like in the tumor or in the cancer case, we can target that point with the help of Niosome. Like anti-neoplastic treatment. In the anti-neoplastic treatment, we can use the Niosomes. In the leishmaniasis treatment, we can also use the Niosomes, and we can also use the Niosome in the delivery of peptide drugs. So peptide drugs these are the large molecular drugs made up of protein, like growth hormone is included in the peptide drugs. We can also use the Niosome for the ophthalmic preparation. So this was all about the Niosomes.